Hello everyone, my name is Shili. I am a fourth-year PhD student from the University of Notre Dame. Today, I am presenting our work, How We Study Aging from a Weighted Dynamic Protein-Protein Interaction Network. We usually call it PPI network. We study human aging. Why? Because human aging is linked to many prevalent and complex diseases, such as cardiovascular disease, diabetes, cancer, and Alzheimer's disease. Even the most recent and widespread COVID-19 seems to be related to aging. According to the U.S. Center for Disease Control and Prevention, around 81% of COVID-19 related death cases are among older people whose ages are above 64. The aging process is also known to be highly influenced by genetic factors. Therefore, it is important to study aging by identification of aging-related genes and their pathways. Traditionally, the aging-related genes are identified via wet-lab experiments, which can be expensive and time-consuming. Therefore, our goal here is to identify aging-related genes computationally, which is much cheaper and faster. Computational approaches for our goal can be categorized into three directions. The first direction is to use aging-specific gene expression data alone. Approaches from this category predict a gene as aging-related if its expression level varies with age. While these approaches do capture aging-specific information, they ignore interactions among genes. However, the aging process, just like any other biological processes, is carried out by genes' protein products that are interacting with each other in complex ways. To overcome this, the second direction is to use PPI network alone. Approaches from this category predict a gene as aging-related if its position in the PPI network is similar enough to the network positions of known aging-related genes. However, current PPI network data is static and context-unspecific, meaning that the PPIs in the network are obtained from different contexts, such as diseases, tissues, patients, and etc. In our study, this means that the PPIs span different ages, meaning that they are not age-specific. The third direction is to use both aging-specific gene expression data and the context-unspecific PPI network data in the late stage. In particular, approaches from this category still use gene expression data alone to identify aging-related genes, and then they use PPI data alone simply to see which of the aging-related genes are linked to each other. So, such approaches still study the two data types in isolation from each other. In other words, they fail to integrate the two data types before predicting aging-related genes. Instead, unlike any of the existing work I just talked about, our goal is to truly integrate the gene expression data and the PPI network data, and to do so in the early stage before we make any aging-related gene predictions. Our early work has already integrated the two data types in the early stage to infer a dynamic aging-specific subnetwork. See the first reference below. In particular, for a given age, we used a network propagation method called netwalk to propagate the gene expression levels onto the entire context-unspecific PPI network to assign the age-specific weights to the PPIs. Netwalk does not have a procedure of extracting an age-specific subnetwork from a weighted PPI network. So, we define our own way to do this. Specifically, given a weighted PPI network, we applied a meaningful weight threshold to treat all PPIs with weights above the threshold as equally important for the given age, and treat this set of age-specific PPI as the age-specific PPI subnetwork. We repeat this process for each age and get a static and unweighted age-specific subnetwork snapshot per age. Finally, we combined these snapshots and formed a dynamic aging-specific subnetwork. This dynamic aging-specific subnetwork is thus unweighted. Our work, which was the best we can do at that time, has advanced the study of human aging. However, there is room to improve even further. First, recall that we need to apply weight threshold when we extract an age-specific subnetwork for a given age. However, it is hard to choose a meaningful PPI weight threshold because there is no clear guideline about which threshold is meaningful. So, one needs to empirically evaluate subnetworks constructed at multiple thresholds, which is computationally expensive. Second, the current dynamic aging-specific subnetwork treats all PPIs with the same importance. It means it assumes that each PPI carries the same amount of aging-specific information. In this case, PPIs that carry different amount of aging-specific information cannot be distinguished. To improve upon our previous work, we propose a novel way to infer a weighted dynamic aging-specific subnetwork. In this way, the aging-specific information can be distinguished by both network structure and aging-specific weight of PPIs. In particular, for a given age, we again use the network method to propagate the gene expression levels onto the entire context-unspecific PPI network to assign age-specific weights to the PPIs. Then, instead of extracting a subset of edges from the weighted PPI network and treating them as an age-specific subnetwork for the given age, we keep all edge from the weighted PPI network, along with the age-specific weights of the PPIs. So, our age-specific subnetwork now has every PPI from the PPI network, where each PPI has an associated age-specific weight. We repeat the process and obtain a weighted age-specific subnetwork per age. This way, by combining all weighted snapshots, we form a weighted dynamic aging-specific subnetwork. Because the weighted dynamic aging-specific subnetwork differs from the entire network in three ways. First, aging-specific. Second, dynamics. Third, weight. To properly examine which component is superior, other than the entire context-unspecific network, the weighted and unweighted dynamic aging-specific subnetwork, we need to have a static aging-specific subnetwork as well. If we show that the best of the aging-specific subnetworks outperform the entire context-unspecific network, that would mean the aging-specific component is superior. If we show that the best of dynamic aging-specific subnetworks outperform the static aging-specific subnetwork, that would mean the dynamic component is superior. If we show that the weighted dynamic aging-specific subnetwork outperforms the unweighted dynamic one, that would mean the weighted component is superior. The problem statement here is, given an aging-specific subnetwork and a set of aging-related genes as input, we first summarize the position of a node in the network into the gene's feature. Second, we use supervised classification via five-fold cross-validation to learn network position patterns of the known aging-related genes and rank and predict the other genes as aging-related based on their feature similarities to the known aging-related genes. Then, we predict top K genes as aging-related. Finally, we use precision recall F-score framework to evaluate how accurate are aging-related predictions made from each analyzed network and subnetwork. Intuitively, precision measures out of all predictions how many are known aging-related genes. Recall measures out of all known aging-related genes how many of them are among the predictions. F-score is the harmonic mean of precision and recall. Formally, we aim to test the following hypotheses. First, whether using any of our aging-specific subnetworks is superior to using a context-unspecific PPI network. Second, whether using any of our dynamic aging-specific subnetwork is superior to using its static counterpart. Third, among our dynamic aging-specific subnetworks, whether using our weighted one is superior to using its unweighted counterpart. Here's the summary of our results. The y-axis shows the accuracy score. The x-axis shows the subnetworks, as well as the random approach that we are comparing. The first thing we'll observe is that all networks perform better than the random approach, which means the performance of all networks are non-random. Then, by comparing the best of the three agent-specific subnetworks to the entire network, we can get the effect of agent-specific component. Indeed, it is the weighted dynamic agent-specific subnetwork that performs better than the entire context-unspecific network in terms of AUPR, recall, and F-score, and with a tight score in terms of precision. So, hypothesis 1 holds. By comparing the best of dynamic agent-specific subnetwork to the static agent-specific subnetwork, we can get the effect of the dynamic component. Indeed, it is the weighted dynamic agent-specific subnetwork performs better than the static subnetwork in terms of all four measures. So, hypothesis 2 holds. Finally, by comparing the weighted dynamic agent-specific subnetwork to its unweighted dynamic counterpart, we can get the effect of the weight component. Indeed, it is the weighted dynamic subnetwork performs better than the unweighted one in terms of all four measures. So, hypothesis 3 holds. To summarize again, we proposed a novel framework for the supervised prediction of agent-related genes from a weighted dynamic agent-specific subnetwork. We confirmed that using an integrated agent-specific subnetwork is superior to using an entire context-unspecific PPI network. And using a weighted dynamic agent-specific subnetwork is superior to using a static counterpart as well as an unweighted counterpart. Finally, I would like to thank NSF for funding this work through a career award, and thank you all for attention.